7.6 Connections of carbohydrate, protein, and lipid metabolic pathways. By the end of this section, you will be able to do the following. Discuss the ways in which carbohydrate metabolic pathways, glycolysis, and the citric acid cycle interrelate with protein and lipid metabolic pathways. Explain why metabolic pathways are not considered closed systems. You have learned about the catabolism of glucose, which provides energy to living cells. But living things consume organic compounds other than glucose for food. How does a turkey sandwich end up as ATP in your cells? This happens because all of the catabolic pathways for carbohydrates, proteins, and lipids eventually connect into glycolysis and the citric acid cycle pathways. See Figure 7.17. Metabolic pathways should be thought of as porous and interconnecting. That is, substances enter from other pathways, and intermediates leave for other pathways. These pathways are not closed systems. Many of the substrates, intermediates, and products in a particular pathway are reactants in other pathways. Connections of other sugars to glucose metabolism. Glycogen, a polymer of glucose, is an energy storage molecule in animals. When there is adequate ATP present, excess glucose is stored as glycogen in both liver and muscle cells. The glycogen will be hydrolyzed into glucose 1-phosphate monomers, G1P, if blood sugar levels drop. The presence of glycogen as a source of glucose allows ATP to be produced for a longer period of time during exercise. Glycogen is broken down into glucose 1-phosphate, G1P, and converted into glucose 6-phosphate, G6P, in both muscle and liver cells, and this product enters the glycolytic pathway. Sucrose is a disaccharide with a molecule of glucose and a molecule of fructose bonded together with a glycosidic linkage. Fructose is one of the three dietary monosaccharides, along with glucose and galactose, part of the milk sugar disaccharide lactose, which are absorbed directly into the bloodstream during digestion. The catabolism of both fructose and galactose. 7.6 Connections of carbohydrate, protein, and lipid metabolic pathways. 195 produces the same number of ATP molecules as glucose. Connections of proteins to glucose metabolism. Proteins are hydrolyzed by a variety of enzymes in cells. Most of the time, the amino acids are recycled into the synthesis of new proteins. If there are excess amino acids, however, or if the body is in a state of starvation, some amino acids will be shunted into the pathways of glucose catabolism. Figure 7.16. It is very important to note that each amino acid must have its amino group removed prior to entry into these pathways. The amino group is converted into ammonia. In mammals, the liver synthesizes urea from two ammonia molecules and a carbon dioxide molecule. Thus, urea is the principal waste product in mammals, produced from the nitrogen originating in amino acids, and it leaves the body in urine. It should be noted that amino acids can be synthesized from the intermediates and reactants in the cellular respiration cycle. Figure 7.16 The carbon skeletons of certain amino acids, indicated in boxes, derived from proteins can feed into the citric acid cycle. Credit. Modification of work by Michael Hagstrom. Connections of lipid and glucose metabolisms. The lipids connected to the glucose pathway include cholesterol and triglycerides. Cholesterol is a lipid that contributes to cell membrane flexibility and is a precursor of steroid hormones. The synthesis of cholesterol starts with acetyl groups and proceeds in only one direction. The process cannot be reversed. Triglycerides, made from the bonding of glycerol and 3 fatty acids, are a form of long-term energy storage in animals. Animals can make most of the fatty acids they need. Triglycerides can be both made and broken down through parts of the glucose catabolism pathways. Glycerol can be phosphorylated to glycerol 3-phosphate, which continues through glycolysis. Fatty acids are catabolized in a process called beta oxidation, which takes place in the matrix of the mitochondria and converts their fatty acid chains into 2 carbon units of acetyl groups. The acetyl groups are picked up by CoA to form acetyl-CoA that proceeds into the citric acid cycle. 196 Chapter 7, Cellular Respiration. Access for free at openstax.org. Figure 7.17 Glycogen from the liver and muscles, as well as other carbohydrates, hydrolyzed into glucose 1-phosphate, together with fats and proteins, can feed into the catabolic pathways for carbohydrates. Evolution connection pathways of photosynthesis and cellular metabolism. The processes of photosynthesis and cellular metabolism consist of several very complex pathways. It is generally thought that the first cells arose in an aqueous environment, a soup of nutrients, possibly on the surface of some porous clays, perhaps in warm marine environments. If these cells reproduced successfully and their numbers climbed steadily, it follows that the cells would begin to deplete the nutrients from the medium in which they lived as they shifted the nutrients into the components of their own bodies. This hypothetical situation would have resulted in natural selection favoring those organisms that could exist by using the nutrients that remained in their environment and by manipulating these nutrients into materials upon which they could survive. Selection would favor those organisms that could extract maximal value from the nutrients to which they had access. An early form of photosynthesis developed that harnessed the sun's energy using water as a source of hydrogen atoms, but this pathway did not produce free oxygen, anoxygenic photosynthesis. Another type of anoxygenic photosynthesis did not produce free oxygen because it did not use water as the source of hydrogen ions. Instead, it used materials such as hydrogen sulfide and consequently produced sulfur. It is thought that glycolysis developed at this time and could take advantage of the simple sugars being produced, but that these reactions were unable to fully extract the energy stored in the carbohydrates. The development of glycolysis probably predated the evolution of photosynthesis, as it was well suited to extract energy from materials spontaneously accumulating in the primeval soup. A later form of photosynthesis used water as a source of electrons and hydrogen and generated free oxygen. Over time, the atmosphere became oxygenated, but not before the oxygen released oxidized metals in the ocean and created a rust layer in the sediment, permitting the dating of the rise of the first oxygenic photosynthesizers. Living things adapted to exploit this new atmosphere that allowed aerobic respiration as we know it to evolve. When the full process of oxygenic photosynthesis developed and the atmosphere became oxygenated, cells were finally able to use the oxygen expelled by photosynthesis to extract considerably more energy from the sugar molecules using the citric acid cycle and oxidative phosphorylation.